Good morning, everyone. I guess everybody here knows me. I'm the Oyedele Adeyi, AP. And so thank you to Dr. Kojo Elenitoba-Johnson, who we have been trying for the last 5 years to get to come and give us a grand round. And that just tells you how busy he is. So we finally nailed him down, and we were hoping he would be in person, but he still managed to escape that. So hopefully in the future we'll be able to do that. But without much ado, I believe many of us know who Dr. Johnson is. So right now he comes to us from the Memorial Sloan Kettering in New York, where he is the current inaugural chair of the Department of Pathology and Lab Medicine and the James Ewing Alumni Chair of Pathology. Right there. Before then, he was at UPenn from 2015 to 2022, and he was at the University of Michigan before that, where he held a lot of leadership positions. Dr. Johnson is well recognized in lymphoma proteomics, and he has contributed well over 180 peer-reviewed manuscripts and numerous chapters and textbooks. His research is supported currently by 3 active R01 grants from the NIH. But there's so many things Kojo is known for, but I think one of the most important aspects of his work has been in lymphoma proteomics, where he's a pioneer. And he's going to be talking today about anaplastic large cell lymphoma. And when I went through his publications, I see that he's got almost 30 papers just on that topic alone. So he is a leader in precision and integrated diagnostics, and his work is quite notable for elucidation and identification of targetable genetic alterations and the underlying pathogenesis in specific lymphoma subtypes. I don't have time to go through his many awards, but I think one of my own personal favorites is the one with his Ramsey Cochran Young Investigator Award, which I think set the pace for a lot of things that Kojo has achieved, because this was a very prestigious award that is given to people under the age of 40. So this was by the United States and Canadian Academy of Pathology. So it's got so many. And again, if for those of you who are interested, I can share with you what I have here right now. So he is also a very active leader internationally and nationally. So he's had a lot of roles at the American Society of Hematology, where he was the chair of the scientific committee of hematopathology and clinical lab hematology in 2015. He was on the board of counselors for the National Cancer Institute at the NIH from 2013 to 2018. He was also president for the American Association of Molecular Pathology from 2017 to 2018. And the list just goes on and on. But I'm just very excited that Kojo is going to be talking this morning about one of his very favorite topics, which is on anaplastic large cell lymphoma. So thank you very much, Kojo. I'm going to share your slide now. And let me see. I think this is the slide, right? Okay. So over to you.
All right. Can everybody hear me?
Yes, I can hear you.
Okay, wonderful. Thank you very much for the opportunity to speak with you. Because Delia has control, I will ask him kindly to help with advancing the slides. I have to start first from a position of apology that I really was looking forward to the opportunity to be in Minnesota in person, but all kinds of conflicts emerged. But I'm going to say this is not going to be the last time I'm going to speak with you. I'm particularly thrilled about the prospect of meeting trainees and helping describe how our journey has evolved in understanding lymphoma and how this could be helpful for them in their own careers. The next thing I have to do is to thank Oyedele Adeyi for putting this together. My friends in Minnesota who run into me in meetings and ask, come and visit. And for now, it's just going to be this talk. But Beyond this, people should feel free to send me emails if they have any questions. Typically, I respond within 24 to 36 hours, so be patient with me. So the topic of today's lecture, which was in the first slide, is anaplastic large cell lymphoma. Next slide, please. These are my disclosures. They're not relevant to the presentation. Next slide, please. What I'm hoping that we accomplish today is that we review the history and evolution of our understanding of the genetic basis and classification of ALCL, understand how precision therapies impact ALCL, and I'll discuss some of the translational research that's emerging from our laboratory, leveraging proteogenomics and better understanding of ALCL and identifying new ALCL entities. So just so everybody knows where they are in the presentation, I'm going to follow this outline. So I'm going to have an introduction that will include a history of the evolution of ALCL as an entity. I'll talk briefly about proteogenomics, or what we like to interpret as proteogenomics, and the tools that are used in that enterprise. And I'll use the opportunity to discover novel oncogenic drivers and targetable pathways in ALCL to illustrate the power of that platform. And I'll summarize and offer some conclusions. Next slide. Next slide, please. Okay, next slide. So lymphoma continues to be an important disease. It's still ranked both in males and females as one of the top causes of cancer deaths. Next slide, please. And generically speaking, as you all know, being pathologists, your sophisticated audience, it's Hodgkin lymphoma or non-Hodgkin lymphoma. That's the major categories. And amongst the non-Hodgkin lymphomas, they generally tend to be classified under B cell and T cell neoplasms or mature/T and K neoplasms. And these are separate and apart from precursor neoplasms. Next slide, please. Whereas the B cell lymphomas account for the vast majority of neoplasia in the Western Hemisphere, and in Europe, the T cell neoplasia are much less represented, account for about 15%, and most of these come from Asia and in Latin America, where they comprise about 35 to 40% of tumors. Next slide. And if you were to break that down even further, the peripheral T cell lymphomas, so-called not otherwise specified, account for about a third of these. And ALCLs, anaplastic large cell lymphomas, account for about 20%. Next slide. Now, one of the fun aspects of being a hematopathologist is that the classifications become even more complicated as time goes on. Those who are practicing hematopathologists like to think about that as job security, because these entities end up requiring more and more parameters for their definition. But broadly speaking, amongst the mature T cell lymphomas, according to what's going to be revised in the new version of World Health Organization Blue Book, which is being published recently, but I use the 2016 classification just to anchor how people think about this. That the cutaneous, extranodal, nodal, and leukemic, the entities I'm going to describe are going to be within so-called cutaneous and nodal. And within the nodal, it'll be the ALK-positive ALCLs to start with. Next slide. And as you can tell, there are other forms of ALCL, which include breast implant associated ALCL. Next slide. So anaplastic large cell lymphoma is the most common pediatric mature T cell lymphoma. It accounts for about 20% of childhood lymphoma. In kids, it's an aggressive disease with frequent extranodal involvement. What's important about this is that up until 1994, other than the fact that there's this set of tumors that occur in children, not much more was known about it. Similar tumors occurred in adults, but it's hard for people who are trainees today to appreciate how much in the way of tools we lacked for characterization of this entity, which is why I'm going to spend time on the history and the evolution. Next slide, please. What was important to note is that following the development of the CD30 antibody, which I'll review, which has now become definitional for the anaplastic lymphoma entity, there are two major prognostic classes of ALCL, those that are ALK positive and those that are ALK negative. And as you can see, today, the ALK positive ALCLs actually portend a more favorable prognosis. So making the distinction between these two categories is the big and more important distinction to make. Next slide. So let me tell you a little bit about the evolution of the history of ALCL and how we ended up learning about ALK positive ALCL. So the initial description was following the development of the KEY1 antibody, which is an antibody against CD30. This was originally developed by Harold Stein in 1982, working with an antibody which was to the natural epitope of the antigen. And then somebody described some sinusoidal lymphomas in 1985, again, taking advantage of this antibody. This is the point in the time in which when we would take pictures, and you'll see this, we could only do this for publication purposes in black and white. So this is a world that is unfamiliar to most trainees today. Immunophenotypic characterization followed. And then in 1994, Steve Morris and Tom Look associated the T25 translocation with the MPM malfusion. This was a big paper in Science, which I'll show you. And subsequently, we've learned that ALK itself has numerous translocation partners, in excess of 30 in multiple different forms of cancer today. Polyclonal antibodies against ALK1 were utilized as a diagnostic tool in 1997. And as I showed, the 2016 classification of ALK positive ALCL has been leveraged for precision pathology. Next slide. So it's important to know that at the time, before the development of the anti-CD30 antibody, KEY1, so it was Key was an abbreviation for Kiel, because that's where Harold Stein was working with Karl Leonard. And so it was called the Key-1 antibody. 1 in German is for the numeral 1. And it only recognized CD30 in the if you had frozen sections. So as a trainee, we had to cut some frozen sections to do immunohistochemistry to recognize this entity. On the right panel, what I try to show here is how early disease, the sinusoidal distribution, is beautifully illustrated by this antibody. And the tumor itself has natural histological characteristics, which I will describe subsequently. Next slide, please. Subsequent to the development of the antibodies, it became clear that there were a recurrent cytogenetic alteration associated with this KEY1 lymphoma. And by the way, at that time, they were not even clear that it was a lymphoma. So again, David Mason, who had developed the antibody, that's who Harold Stein worked with to develop the antibody against CD30, working with some cytogeneticists, described the translocations involving recurrent breakpoints in chromosomes 2 and in particular chromosome 5. Next slide. Steve Morris and Tom Look, as I mentioned, now identified the participating genes as NPM and ALK. In 1994, this was a landmark paper because it was the first time anybody showed the products of a recurrent fusion in ALCL. Next slide. So histologically, this is not going to be unfamiliar to this audience. You can see in the top right panel, H&E stains here, the tumor cells themselves are about 50 times bigger than the index normal interspersed lymphocyte. And they're characterized by enlarged nuclei, which have large prominent nucleoli, really abundant cytoplasm. They, according to George Soule and Ben Horok, they have this so-called perinuclear huff. So it's a Golgi intensification in these cells. I'm sorry, I don't have my arrow active, but if you look at the cell right above where the arrow is, you'll see, it's like 2 large nuclei under which there's a third one, which circumscribes the huff. By definition, these are intensely CD30 positive, both membrane And the intensification we see in Golgi, this is and then in the right, you can see a case of NPM ALK positive ALCL, where both there's nuclear and cytoplasmic expression of the antibody. Thank you very much, Dele. Next slide. And as you can see here, you can see the sinusoidal growth pattern. The arrow basically shows you the so-called hallmark cells of Ben Horok and George Del Sol. This is a horseshoe-shaped cell that circumscribes the Golgi, markedly enlarged cell, and this is what is considered the typical ALCL cell. Next slide. So several variants have been described. Some people have become famous with this variant. It actually has a very protean histologic manifestation. Some of them look like sarcomas, so you need a high index of suspicion to detect these. And this is a so-called lymphohistiocytic variant on the left. Next slide. Others have described so-called giant cell variant and small cell variant. Marcia Kinney, who is a very well-known hematopathologist, described the so-called small cell variant of anaplastic large cell lymphoma, which sounds like its own oxymoron. Next slide. They tend to show T cell associated markers. Now remember, T cell lymphomas tend to lose expression of pan-T cell markers, but periodically you might see CD4 expression. But importantly, they express TIA1 and perforin. This is work that was done by Laszlo Kranax when he was a fellow in Elaine Jaffe's lab. And they also show intense reactivity for CD25, which is the interleukin-2 alpha receptor. Back in the day when I was a trainee, the epithelial membrane antigen that was used to recognize these. But this is part of the reason why there was confusion about this neoplasm. In fact, many of the ALCL cases in the past were misdiagnosed as carcinomas because of the large size of the cells and how different they were from typical lymphoma cell. And if you thought it was a lymphoma and you thought it was an epithelial neoplasm and you used EMA and you saw reactivity, well, some people could misdiagnose it as carcinoma. Next slide, please. So what's important to keep in mind is that there are many CD30 positive neoplasms. They're not exclusive to lymphomas. Some germ cell tumors show expression of it. And there are also ALCL positive neoplasms that are not lymphomas. So you have to demonstrate both 30 and ALK, and to the extent that you can, lymphoid markers to be inclusive of a diagnosis of an ALCL. Next slide. Okay, so let me tell you a little bit about ALK. So anaplastic lymphoma kinase is a receptor tyrosine kinase. It is a member of the insulin receptor superfamily, and it really drives neural and brain development, and it silences differentiated cells. In other words, if you see expression of ALK in any mature cell that is not neural, it's likely to be neoplastic, and it's not expressed in lymphocytes. So if you see ALK expressed even with low intensity in lymphoid cells, it's very likely the consequence of a structural alteration that is a neoplasm. Next slide. So the normal full-length ALK signals through its normal ligand is a FAM150 family, and what happens in the translocation is that nucleophosmin, the N terminal of nucleophosmin, gets joined to the C terminal of the anaplastic lymphoma kinase. Next slide. Well, just if you advance that, it should have animated, but not. That's interesting. In any event, that process leads to downstream oncogenesis through suppression of apoptosis, increased proliferation, and activation of pathways that it is a super oncogene. If you express NPM-ALK in mice, they all develop lymphoma. And sometimes the lymphomas may not even just be T-cell lymphomas. They may be B-cell, or sometimes they may even have a sort of a histiocytic phenotype if driven by CD4 driver. Anyway, so I show here Steve Morris, who worked extremely hard to identify translocation, tried very hard to develop a drug against this. Was very, very frustrating because there was no interest from pharma. Remember that NPM positive ALCL accounts for more than 80% of the gene fusions in the ALCL in children. So you would think this is a high frequency genetic alteration that people would be interested in, but it was tough to obtain interest from pharma because it was considered a rare and orphan disease until 2009. Next slide. Next slide. The reason why it became prominent was because people identified that ALK was rearranged in lung cancer. Lung cancer, 220,000 cases a year. This all of a sudden takes it from the spectrum of being involved in an orphan disease to a very common one. And so a drug was developed very quickly. The fusion in lung cancer is the EML4 ALK translocation. And I mentioned that there's more than 30 partner genes that have been described in virtually every neoplasm you can imagine. In fact, I use this opportunity to tell trainees that if you're taking boards and somebody says, which one of these is an oncogene? It doesn't matter where the tumor is located, brain, breast, colon. If you see ALK as one of the options, select it. You'll be correct, even if you don't know the answer, because it's implicated in neoplasms either by point mutation, amplification, gene fusion, in neoplasms virtually every organ. So in 1994, as I've told you, the T25 was shown to contain the NPM ALK. Next slide, please. Now we have more than 30 in different neoplasia, different partners of ALK, all driving ALK from the N-terminal, as you can see. So I don't think that these have been exhaustively characterized. When I say exhaustively, there's always going to be a new partner. So this might be an opportunity for a resident to become famous if they can find yet another partner of ALCL. And you can see that it's involved in many different kinds of neoplasm, inflammatory myofibroblastic tumors, even diffuse large B-cell lymphomas, etc. Next slide. Okay, so one way in which you can recognize that you have a nuclear fusion partner as demonstrated with NPM, where you see the intense nuclear and cytoplasmic reaction for anti-ALK. As it turns out, NPM is the most prominent nuclear fusion partner. All the other fusion partners are in different cellular subcompartments, so they have different patterns. So this is very distinctive for NPM-ALK fusions. And NPM, the nucleophosphate itself, is a nucleolar phosphoprotein, which is a shuttle protein. It shuttles proteins from the nucleolus into the nucleoplasm, in the nucleus, and into the cytoplasm. So you'll see expression wherever NPM is. Next slide. In other invariant translocations I mentioned, you can see nuclear and cytoplasmic in the left panel, and you can see exclusively cytoplasmic. This is a TPM3 ALK fusion I'm showing on the right. Next slide. And this is an inflammatory myofibroblastic tumor. This is, that has all these variant fusions I showed to the left. And here you can see no nuclear signal and plenty signal in the cytoplasm. Next slide. And this is the pattern you would see in lung cancer. So up top, I show you histology. And the bottom left, I show you FISH, which is a split probe approach here, basically showing the EML4 ALK fusion. And immunohistochemically, you can see lung cancer expressing ALK. You can see the intensity is less and there's no nuclear signal. Next slide, please. The consequence of knowing that ALK is a super oncogene, necessary and sufficient to drive oncogenesis, especially in the context of lymphoma, made it possible for folks to now develop small molecule inhibitors of ALK, and in clinical trials, just extremely impressive remissions, which have been persistent. So I show here in a paper published by Yale Mose, Megan Lim, and others, where they demonstrated that targeting ALK with crizotinib, which is an ALK inhibitor, an ALK c-Met inhibitor, led to durable molecular suppression of detection of NPM-ALK fusions. This is really remarkable because this basically credentials this as a precision therapy. And that's consequent to the discovery of this translocation. So this is a very, very fulfilling result, frankly, from all the history I've told you to the discovery of creating a drug that has this degree of specificity. And you can see, I don't have strong credentials as a radiologist, but trust me, nonetheless, if I tell you that there's a remarkable decrease in disease burden post-cycle, even just after 28 days, and histologically, you could see the marked depletion of any signal of ALK in the biopsy materials obtained. Next slide. So just to summarize, the first part of the presentation, ALCL, ALK positive is a distinct clinical pathologic entity, okay? ALK positive ALCL T/NK in children and adolescents and young adults carries a favorable prognosis. I think I showed you that earlier on. And especially with new treatments, the quality of life is very high because of the effectiveness of the drug, with or without adjusted chemotherapy. And ALK and pathway inhibition is obviously a viable therapeutic strategy in ALCL. So I use this as an opportunity to explain to trainees and pathologists how important what we do is. When I was a trainee, we were almost compelled by clinicians to basically just have 3 categories of lymphoma. That was enough. And some people discourage the kind of observational scrutiny and specificity that pathologists bring to the table. Absent that, we wouldn't have recognized that this was a special entity that actually has its own specific kind of treatment and has implications beyond lymphoma into lung cancer and every other thing. So continue with the commitment to recognizing when you have something that looks different. And in particular, if it behaves different in a different demographic, you'll be on to the specific characteristics that make it possible for people to develop targeted therapies. And I think this is a good story about how being a fastidious pathologist led to the characterization of a distinct clinical pathologic entity with distinct treatment implications. Next slide. Okay, so I've talked to you about ALCL, ALK positive. I'm not going to talk to you about ALCL, ALK negative, which I said for the most part has a heterogeneous prognosis. And in that category, I'll just talk about the cutaneous, so-called primary cutaneous CD30 positive T cell disorders and primary cutaneous ALCL. Next slide. I mentioned that, so among the ALK negative ALCLs, their prognosis is worse, but within that category in the top, in the bottom right, you can see that you could even still further subdivide them into ones that are bad and even worse. And others, Andy Feldman at Mayo, characterized structural alterations involving DUS22-IR4, TP63, and we embarked on a study to further distinguish new alterations in that disease. So this story is one of such a discovery process leveraging proteogenomics. Next slide. Okay, next slide. So our laboratory leverages genomics, which basically scales the central dogma. So DNA, as you remember, gets transcribed to RNA, which ends up being translated to proteins. We interrogate these at scale using primary lymphoma samples. So if there's a translocation point mutation, we should see gene fusions using RNA, and we have deep domain expertise in mass spectrometry-driven proteomics. If we're compelled by the hypotheses that we see from these preceding approaches, we'll do functional studies, and we will make animal models, which I won't get a chance to talk to you in any detail about today. But ultimately, our work is driven by our observations from primary clinical samples, which I have boxed in red here, And unless those observations are validated in primary clinical samples, we don't find it to be a satisfactory execution of the project. Next slide. So everybody knows about genomics, but people know less about proteomics. So proteomics is the large-scale study of the proteome. Mass spectrometry can unambiguously identify proteins. It's unbiased. In other words, you don't need preconceived ideas of the protein you are looking for. And very importantly and powerfully, it can identify the precise site of post-translational modifications. Next slide. And you want to study the proteome because we only have just under 24,000 genes. I'll remind you that the worm C. elegans has 19,000 genes. Okay, so we don't have much to brag about, right? In comparison to the worm that just creeps and doesn't have what you would consider to be sophisticated organs and so on. So there's a lot that's being contributed by the epigenome and by the proteome. Next slide. So for out of our 23,000 genes, we encode over 1 million protein isoforms. The strategy we use to interrogate our proteins, which we obtain from our tumors, is by electrospray tandem mass spectrometry. Next slide. This is one version of it. This is a sophisticated piece of equipment, which is now basically a desktop equipment. Suffice it to say that this tool makes it possible for us to identify differences in nominal molecular mass of about the size of a hydrogen atom. That's 1 Dalton. So they've become really sophisticated and we can do really special things. Next slide. This might animate, but if it doesn't, that's actually good. This is an example of what the data looks like. What I just wanted to offer here is that if you see highlighted in red, there are amino acid sequences. There's just 2 peptides here that unambiguously identify a protein here as STAT3. The reason I'm showing this is that unlike in genomics, you don't identify the entire open reading frame of a gene. Just 2 unambiguously identified peptides can be used to unambiguously assign identification to a protein. Next slide. Okay, so using this approach, on a bad day, we can identify 6,000 proteins from any sample that we interrogate. So we're in the realm of doing whole proteome analysis where I know nothing about the sample. If I have it analyzed in the mass spectrometer, I can retrieve, on a bad day, north of 6,000 proteins. Next slide. Also, everything I've told you is what you do when you want to identify a protein. What I'm telling you now is that you can even identify post-translational modifications like phosphorylation. And as you know, phosphorylation is a post-translational modification that is added to proteins by kinases, which are highly overrepresented as oncogenes in the human genome. So one way we conceived of identifying new oncogenic drivers that were kinases was leveraging the mass spectrometry technique, looking for overrepresentation of phosphorylation. Next slide. And so again, in an unbiased way, the same sample that I interrogate that shows me 6,500 proteins, I can look for phosphopeptides and identify more than 17,000 phosphopeptides, unbiased, with site-specific localization. I can tell you exactly which residue is being phosphorylated, serine, threonine, and tyrosines, just in an unbiased way. Next slide. And it's highly reproducible. That's the punchline of this slide. So the R values for repeated experiments are north of 0.99, which is really, really impressive. Next slide. So if you've ever done one of these experiments, you get a gigantic Excel spreadsheet, right? If you're getting 17,000 phosphopeptides, sometimes it's too difficult for Excel to manage, right? In fact, crashes. So there's specific software that people use to analyze this data. But what I'm trying to show you here is that if you just use the sort function and ask, okay, I have analyzed this sample by phosphoproteomics, I want to know which protein exhibits the highest levels of phosphorylation just by spectral count or by total number of peptides. And here, just by sorting in the rightmost column, we observe ALK. So if you display this data as a graph, next slide, please, it looks something like this. This is this graph basically shows that there are several peptides that make ALK an outlier in the degree to which it is phosphorylated. It is hyperphosphorylated. You remember that I said with receptor tyrosine kinases, if they're oncogenically activated, what typically happens is they first autophosphorylate themselves before they do any downstream signaling. So taking advantage of this, we thought in an unbiased way, we could discern what an oncogenic tyrosine kinase that was unknown that is driving a phosphorylation process in a tumor sample could look like. So we repeated the experiment using other samples. Next slide. If you do this using a CML sample, the outlier protein is ABL. Next slide. If you do this using an ALK-positive ALCL, the first one I showed you was a lung cancer that was EML4 ALK. You can see ALK here being a giant outlier protein. So this basically tells you that the driving phosphorylation event is phosphorylating ALK, and the most likely explanation is an alteration involving ALK. Next slide. So we said if this is true, we ought to be able to use this to discover other oncogenic drivers. More importantly, I could use that entire data and create a heat map to see what happens when I inhibit ALK with site-specific specificity and localization. Next slide. Okay, so now that I have this technology in hand, can I use this to identify novel oncogenic drivers? So Delphine Roland, who was a postdoc in the lab, but now she's has her own lab in Strasbourg in France, wanted to take advantage of this to identify oncogenic driver kinases and develop the concept of outlier kinase profiles. Next slide. So we decided to use that to address the issue of unknown kinase fusions in CD30 positive lymphoproliferative disorders, which are the second most common group of cutaneous T-cell lymphomas. The two different types is a cutaneous anaplastic large cell lymphoma and so-called lymphomatoid papulosis. So no consistent genetic alterations have been described affecting kinases. And these have intense reactivity for CD30, as I show in the bottom right, okay? And how these are different genetically from anaplastic large cell lymphoma, ALCL positive. Andy Feldman had shown that there were some structural alterations involving DOS2, IRF4, and TP63, but really kinases hadn't been implicated. Next slide. So we did this outlier kinase analysis, which you've now become experts at because you've seen examples of it. And then we identified this protein called TYK2, which is tyrosine kinase 2. So, you know, even Daley has become an expert. Right now, he could identify that this was an outlier kinase, right? And this myelin cell line is supposedly obtained from a cutaneous T-cell lymphoma. So we were highly suspicious that something's got to be going on with TYK2. Next slide, please. So we did RNA sequencing, right? Because we're looking for a genetic basis for that outlier signature. Next slide. And Mark Keel and Thiru Velusamy identified the NPM TYK2 fusion. I should tell you a little bit about TYK2 coming right up, but you now recognize NPM, the same NPM that is implicated in the fusion with ALK. Thiru cloned the translocation and sequences, which I show on the bottom using Sanger at the DNA level. So we knew it was a DNA-based event that was responsible for the RNA fusion. And this led to the identification of this new translocation, the T519, in these cutaneous ALCLs. Next slide. And so basically this is work done by Thiru Velusamy in the lab, where he cloned the breakpoint. And you can see in the HH cell line to a different cell line, there's no band there, and the sequencing basically showed that NPM1 TYK2. Next slide. So we made FISH probes, both direct fusion and break apart, and we analyzed lymphomatoid papulosis, primary ALCLs, and some other peripheral T-cell lymphomas. Virtually 17% of the CD30 positive cutaneous lymphoproliferative disorders and ALCLs carried rearrangements involving TYK2. Some of the rearrangements we saw by break apart, and only a few by NPM TYK2 direct, which told us that TYK2 had other fusion partners other than NPM, just like we've observed with ALK. So the architecture of the fusion is schematized here. The N-terminal of the fusion co-opts the oligomerization and histone binding domains of NPM, and the C-terminal of TYK2, similar to what we see in ALK. Next slide. Normal TYK2, I should tell you, is the original JAK kinase, okay? In fact, its name is called TYK2, meaning tyrosine kinase 2, which is sort of an unfortunate nomenclature because it seems like it's second to something else. Well, basically it was second to something like ABL back in the day when they didn't know much about tyrosine kinase, but it's the founding member of the JAK family, and nobody's really studied it in great detail because nobody found structural alterations in any form of primary human cancer. So the NPM1 TYK2 fusion was the first gene fusion that was demonstrated in primary human samples to be affecting TYK2. Prior to that, people just knew it was a kinase and they didn't really pay much attention to it. On the other hand, its sister cousins like JAK1, 2, and 3, JAK2 is famous for being recurrently mutated in the chronic myeloproliferative disorders. Because of this, JAK2 has been more or less the most prominent JAK kinase. But the founding member of that family is TYK2. What's important is that they are downstream of cytokine receptor signaling. Sometimes they heterodimerize with one another, but they all activate STAT signaling downstream. Next slide. And so here are some Western blots basically showing that if you use an antibody on the left against NPM, in the myelin line that carries the NPM1 TYK2 fusion, you'll see a unique band at about 100 kilodalton corresponding to the chimeric fusion only in the myelin line, which basically tells you that this is the only line out of these T-cell lines that carries this fusion. In the middle panel, you'll see if you use an antibody against TYK2, you'll see NPM1 TYK2 and degradation products only in the myelin lane. Similarly, phospho-TYK2 is only demonstrated in the myelin line. And the reason this is important is that this is an exclusive alteration showing that kinase signaling is being driven in these samples. And on the right, what's important to note is that the phosphostats are activated in the myelin line in comparison to JERCAT, for example, where you see no signal whatsoever. Next slide, please. This is what you see if you do immunohistochemistry for phosphostat 5, which is downstream of TYK2. It is highly expressed in nuclear signal in the CD30 positive lymphoproliferative disorder, which I show on the right, whereas on the right, there's much less signal in the CD30, in the NPM1 TYK2 negative ALCL. Next slide. What I try to show here is that this signal, if you just look at the top segment on the left, we created a kinase dead. This is the K462R. On K462R is kinase dead. So if you eliminate the kinase activity, you abolish the phosphostat 1 signal. And that's the same thing with phosphostat 3. It's the same thing with phosphostat 5. In other words, the kinase activity is critical for the signaling, which means if we develop a kinase inhibitor against TYK2, then we can shut down this pathway through which it drives oncogenesis. Similarly, on the right, I show using a transcriptional reporter assay that the kinase activity is required to be able to transduce signals through the STAT family, STAT 1, STAT 3, STAT 5. Next slide. So On the right, what I show is that if we silence TYK2 using shRNA, two different shRNAs, that's in red and in blue, we can inhibit the growth of the myelin cell line in comparison to the vector. We can significantly inhibit the growth of the tumor. So I'm showing genetically that by silencing TYK2 or by expressing the kinase dead mutant, we can not just only shut down the biochemical pathway, we can also inhibit the growth of the cells in vitro. Next slide. Similarly, lines that have any kind of JAK activation, so TYK2 and the JAK family kinases have significant homology, and despite the claims that the drug was a highly selective inhibitor against TYK2, it affected all of the JAK kinases, what I showed. But the IC50 was close to very similar for all of the tumors that carry JAK mutations or NPM1 TYK2. The HH cell line that had no JAK alteration or TYK2 alteration was basically resistant to treatment, which is the black line we have at the top. Next slide. When we xenograft the myelin cell lines into animals, this Anaxa Hasarbude in the lab created lines that carried luciferase so we could monitor them by in vivo imaging. The tumors are much smaller, so there's a significant reduction in the tumor volume if you treat with the TYK2 inhibitor. Next slide. So to summarize the second part, recurrent TYK2 fusions in cutaneous ALCL were demonstrated by my lab. We demonstrated that NPM1 is one of the fusion partners. The NPM1 TYK2 transfusion results in constitutive activation of TYK2 kinase and STATs, as I tried to show you, and we're making the case that TYK2 may be a novel therapeutic target in a subset of ALK-negative ALCLs. Next slide, please. So Mark Keel and Thiru became famous for this discovery, but what I typically tell them is that it's more important. It's nice to make a discovery. It's more important that it is true. So for 7 years after we described this, I never saw an NPM1 TYK2 fusion again, which was very concerning. That did we find some kind of a strange aberration? But others had reported structural alterations associated with TYK2. Next slide. So my friend Jojo Ingarami at Cornell across the street identified other fusions affecting TYK2. PABC4, which he reported months after we reported our own observation with NPM1 TYK2. Next slide. And then my friend Andreas Rosenwald also reported NPM1 TYK2 fusions occurring in cases of mycosis fungoides. Actually, what was interesting about this is that this basically shows you a patient in which they saw a rash, which is sort of nondescript, and then it sort of got worse and subsequently seemed to get worse. But what was unified for all of these three lesions, which, by the way, at initiation, they didn't consider to be a neoplastic process until it became what I showed you on the right. What unified all of these lesions was the presence of NPM1 TYK2. So this seems to be its own clinical pathologic entity, which can be difficult to pick up at initial diagnosis. And some of the cases of mycosis fungoides is somebody trying to give me a signal here? From what we have come to learn is that this gene fusion may characterize a distinct clinical pathologic entity outside of the spectrum of so-called cutaneous ALCL or CD30 positive lymphoproliferative disorders. I should mention that within a year of arriving at Memorial, we've now identified 7 different translocation partners of TYK2. And importantly, these occur in systemic ALCLs or peripheral T-cell lymphomas that are ALK negative. So this is not just a cutaneous disease, and we're studying the biology of it. Next slide. So now I box in red here that we now have this category of TYK2 rearranged ALK negative ALCLs, and we're trying to understand the biology of it, how they behave biologically, so we can determine the extent to which this is its own entity. Next slide. So to summarize this part, recurrent TYK2 fusions occur in cutaneous ALCL, NPM1 fusion partners. I mentioned that constitutive activation occurs through STAT, and that may be a novel therapeutic target. Next slide. And to conclude, next slide, ALCL itself is a heterogeneous neoplasm. Orphan neoplasms, I think it's important to continue to study. So even if you think that there's not a large number of people who have the tumor you're interested in, I think it's worthwhile continuing to study because it offers insights into other diseases that there's no way you can know ahead of time. And that becomes important in common cancers. I think that it's important to underscore that pathologists continue to play critical roles in characterization of disease and contribute novel insights to disease pathogenesis. A mechanistic understanding of disease pathogenesis eventually leads to development of precision oncology therapies, and this trajectory can be replicated in multiple other cancers. Next slide. Oh yeah, so as you all know, I don't do much work. I depend on the effort, intelligence, and commitment of my team. Of course, the brains of our operation is Dr. Megan Lim, who many of you know is my boss at work and particularly at home. Jack Liang, who came to us from Austria, came with us from Philadelphia to New York, who's been doing a lot of the NPM1 TYK2 work, is characterizing the mouse. My colleagues at the University of Pennsylvania, my colleagues at the University of Michigan, and the government, of course, for funding support. Next slide. Thank you so much for your attention. I'm happy to take questions. And maybe we can...
Thank you very much, Kojo. And just moving the slides, it's like I was tempted not to move faster than you because I really wanted to see what comes next. So that was quite interesting. We have about 5 minutes for questions. Anyone?
I have a question. This is Ishmael.
Ishmael, yeah.
Yes, I know you haven't talked about ALK negative ALCL, but I'm pretty sure you have more information about it than me. I was wondering what there are a subset of cases of classic Hodgkin lymphoma that express cytotoxic T cells, and those cytotoxic T cells are actually negative for PAX5. So how can you make that distinction between ALK negative ALCL, especially if they don't have like DUS22 or JAK2 or TP63? And also, that's another part of the question is, I've read like in a paper that it wasn't very recent, but about like they use CCL22, CD83, and STAT3 and TUBB2B as like an IHC panel to differentiate between classic Hodgkin lymphoma from ALK negative ALCL. So I was wondering if you have any experience with that and how would you make that distinction?
Okay, so let me unpack your question because it was a long one. Actually, there was 2 questions, and in the second question, it's a multi.
Your voice is a little bit low.
Really?
Yes, now it's fine. Yes.
Okay, sorry about that. I said that I have to unpack your question because there was 2 questions, and the second is a multi-part question. As far as Hodgkin lymphomas that don't demonstrate, that sort of sit in the boundary between ALCL, this is a controversy that has existed forever. I just have to basically say that you use every tool available to you, including clonality studies. It's unusual for Hodgkin lymphoma, according to its current definition, to show strong T-cell clonal restriction or monoclonality. We have come to depend on using next-generation sequencing to pick this up because, as you can imagine, it affords you determination of CDR3 sequences when the T-cell receptor. This is different from running it on a gel. So that's why we use NGS. If you're really desperate and you really need to make that distinction. Of course, the absence of the pan B cell markers and the transcription factors that characterize B cells are also helpful in that distinction. But the clinical picture is also very important. It can be a challenging distinction to make, but the recommendation is that you leverage every tool available to you. Your second question is sort of related because every now and then you find somebody reporting that there is a new protein biomarker that distinguishes one category of diseases from another. What we've learned is that hanging your hat on one antibody is likely, a single parameter characterization is likely to mislead you. You want to look at the entire picture, including the panel. And by the way, you will get to a point where, when I was a trainee, Hodgkin lymphoma never expressed CD20. And when we started getting better at immunohistochemistry, we started seeing some weak signals here and there, variable signals, and then somebody now accepted that this could be part of I'm talking about classic Hodgkin lymphoma, as opposed to NLP. So use a panel-based approach, use the entire picture to come to a conclusion, and if you're in deep trouble, I would go to the genetics.
Wow. So, yeah, thank you very much. So this is what we are going to do. We are going to stop recording now. For those who have to leave, please feel free to leave. But I think I want to entertain the other questions. So that means we run ahead of our time. Sorry about that, Kojo. I hope you don't mind. So if you have to leave, we can leave. And Bill can stop recording. Yeah, Bill can stop recording now. Mihao.
Yeah, thanks, Dr. Elenitoba-Johnson, and thank you shared the legendary story of the gene fusion in the ALCL. And so I have two questions. One question is that right now, whole genome sequence is not uncommon to be standardized practice in the some cancer cases. How far do you think the whole protein sequence will be practicable in the clinical practice? That's my first question. My second question, so your whole career is focused on gene fusions, but as we know that if you look at the gene fusion, it's not a random process. In the molecular level, two particular genes have to be physically close each other and have to break down to make two breakpoints at the same very precise intron, and then they fuse together. Otherwise, you are going to cause a lot of problem. And also in the disease-wise, we know it's more like lymphoma, sarcoma, but also in certain of carcinoma. But what's the exact mechanism of this gene fusion occurs? I still think it's largely unknown. Do you have an insight for that? So that's my second question.
Okay, I'll answer your second question first. So remember that lymphocytes have the capacity naturally, physiologically, to rearrange their antigen receptor genes. Sometimes, aberrantly, that mechanism gets extended to other genes. So in B cells, not only that, there's a somatic hypermutation process in B cells that leads to a activation of AID, which can lead to all kinds of genomic alterations which are problematic for the cell. It's a mistake.
Yeah.
And T cells don't have somatic hypermutation. If you discover one, you'll get a Nobel Prize. So there is not that mechanism, but they still have to rearrange the antigen receptor genes. And so most of the gene fusions that we found occur in the T cell lymphomas. I think that process itself is separate from the process that's required for gene fusions. I think that, remember that there's a lot of splicing related joining. Actually, we've discovered some trans-splicing alterations that occur at the splicing level. But what makes it able to be propagated really is at the genomic level. And I don't think that process is completely understood. But I think it is a perversion of the physiologic process that move huge chunks of chromosomes together that leads to this kind of an issue. So your first question is that at some point whole genome sequencing will become sort of routinized clinically. I suspect I will be alive in my career to see that occur. But proteomics, when will that occur? Well, so I know you know this, that at least from the mass spectrometry-driven process, virtually all top caliber microbiology labs use mass spec for their bacterial identification. In fact, even sensitivity determination. So that's one application that's occurring in high throughput in lab. Second area, and we're doing some work on this actively, is in the detection of serum paraproteins. So, you know, if you look at a person who has, for example, a plasma cell dyscrasia or multiple myeloma, there's people in chemistry who do immunofixation, and maybe they will do serum protein electrophoresis and sign them out. It starts becoming problematic these days because there are drugs like daratumumab and so on and so forth that contributes to that peak. So being able to discriminate the drugs from the paraprotein that is being produced by the tumor requires the sophistication of mass spectrometry. And we see that area as an area that's going to explode because every day you look up, there's a new drug that is an antibody-based therapy. So that's going to be, at least in the next 5 years, an area that I think will be explosive for the application of mass spec-driven proteomics clinically. And third, in your same state, and here we utilize mass spec routinely for the detection and typing of amyloid.
Thank you.
Yeah. So we'll take two quick questions, one from Zoe, and then I think Marnush, you have a question, right? So Zoe.
Hi, good morning. It's a fantastic presentation. One of the questions I had, I don't know if you showed them, maybe I missed it, but what was your enrichment level for the phosphoproteome? And normally the phosphorylation events are mainly driven, especially for phosphotyrosine kinases, are mainly driven by phosphorylated tyrosine sites. So what was your enrichment level for that, given that that's kind of like one of the major drivers of signaling during signal transduction?
So that's a very sophisticated question. So the stoichiometry for phosphorylation is very low. For phosphotyrosines is 200 times even less than serines and threonines. The protocol that was used, I didn't get into the chemistry. There's one in which we use immobilized metal affinity chromatography, or IMAC, to enrich for the phosphopeptides. And then it used a triple antibody-based enrichment for phosphotyrosines. After you've done the enrichment for IMAC to be able to detect the low stoichiometry phosphotyrosines. And that's what generated the data that I showed you for the ALK antibody. So it's in the order of greater than 500 to 1,000 times the basal level.
Awesome, thank you.
Thank you. Thank you.
Thank you. So we'll take one more question, Marnush.
Sure. Thank you so much. It's such a pleasure to have you as a speaker. I am already looking forward for the next talk that you will have in person at you.
Yeah, sign me up.
For sure, for sure. I have two questions, and then they're more of a morphologic. So you mentioned about the sinusoidal pattern of ALCL that is known from really old time. Right now in my practice, when I have ALK negative ALCL, which is a scary thing for me, I rely on it. But I don't know, like you have much more experience with ALCLs. Should I really rely on it? Should I be more flexible? And then when I see, scattered cells, which are strongly CD30 positive, I should consider like ALCL. And is there any biology behind that, sinusoidal pattern? Are these like stick together and travel? That's one question. The other question for TYK2 rearranged cases, did you by any chance see any subtle morphologic difference with others that maybe we can use?
Great questions. I'll answer the second question first.
Sure.
TYK2 rearranged ALCLs can be very subtle. In fact, as I showed you from the cases that were reported by Andreas Rosenwald and Katia Maros, even grossly in the patients there might not even be a prominent rash. So it's very subtle, and the neoplastic cells or the infiltrate itself may be more or less inflammatory, and the atypia is not really very staggering. So you need a high index of suspicion. And what we've done at Memorial is just to make a fish probe protocol. Whenever it is we have a suspicion, we do the DUS22, TIC2, JAK2 probes in the ALK negative cases. That just helps you, give you some peace of mind. With regard to sinusoidal distribution of ALCL, well, some other lymphomas actually have a sinusoidal distribution. When I was a trainee, there was an entity known as so-called sinusoidal B-cell lymphoma.
Yes, yeah.
B-cells, yeah. It's only when all of the parameters fit. So it goes back to the initial point that I made. Any single parameter characterization of any entity is likely to be wrong. You want to use a series of parameters to assemble the evidence to properly credential the entity you're describing. So it's a sign, but it's not everything.
Sure. Thank you so much.
Thank you.
Okay.
Thank you.
So on that note, I know we can go on and on and on. And so you see why you have to plan to come back, Kojo. We're looking forward excitedly to see you. I think you have a session with the residents still, right?
I should.
I think so, yeah. Okay, so we are probably taking part of the residence time. So thank you very much, everybody. I put the code for today in the chat, 4916, for those of you that need to claim CMB. See you again. Thank everyone. Bye.